Hello, my name is Joshua Mugabi. I welcome you to Nucleic Acid. Our lesson objectives are nucleotides, DNA versus RNA, nitrogenous bases, DNA structure, types of RNA, Watson and Crick, DNA organization. Nucleotides. DNA and RNA are composed of monomers called nucleotides. That is, DNA and RNA are composed of monomers that are called nucleotides. Now I want us to look at this monomer here, the structure of a nucleotide. This structure of a nucleotide contains a pentose sugar. This one here is our pentose sugar. The whole of this one is the pentose sugar. Then it has the phosphate group. Here we have our phosphate group. And then the nitrogenous base. This is our nitrogenous base. So a nucleotide consists of a phosphate group. It consists of a pentose sugar and nitrogen base. So that's why we are saying that DNA and RNA are composed of a monomer, which is called a nucleotide. The whole of it is a nucleotide. A pentose, that is a 5-carbon sugar, which we have here. It is a carbon 5 sugar. And if you look at it, it has 5 carbons. If you can count, this is the first carbon here, second carbon, third, fourth, and a fifth. That is a pentose sugar. The pentose sugar, it has got 5 carbons. Then we have a phosphate group. Our phosphate group is this one here. This is our phosphate group. And then it has the nitrogenous base. The nitrogenous base is this one here. This is our nitrogenous base. So all those ones, they make up what we call a nucleotide. They make what we call a nucleotide. It is made up of a phosphate group. It has a pentose sugar and it has a nitrogen base. We want now to look at DNA versus RNA. We want to look at the composition of a DNA and RNA can be differentiated by certain key structural characteristics. So we want to look at those characteristics that differentiate the DNA and RNA. We shall start with the pentose sugar. DNA is made up of deoxyribose sugar, whereas RNA is made up of a ribose sugar. Now when it comes to the nitrogenous bases, the DNA has thiamine, whereas RNA has uracil. When it comes to the number of strands, DNA has a double strand and RNA has got a single strand. The basic function, this is the master copy of the genetic instruction, that is the DNA, and RNA is temporary copy of specific instruction. It is just a copy of specific instruction. So those are the differences between the RNA and DNA in certain structural characteristics. DNA, deoxyribose sugar, RNA, ribose sugar. When it comes to nitrogenous bases, DNA has got thiamine, RNA has got uracil. DNA has a double-stranded and RNA is single-stranded. The basic function for DNA, it is a copy of genetic instruction, whereas RNA is just a temporary copy of the specific instruction. Nucleic acid comparison. We want to look at the DNA in terms of the nucleic acid comparison. Now, when you look at RNA nucleotide, we want to compare it with the DNA nucleotide. You will see that all of them, they have got the phosphate group. This one has a phosphate group. So the phosphate group is the same. If you look at their phosphate group, all of them, they have the phosphate group, which is the same. Now, when it comes to the ribose sugar, if you look at the ribose sugar, RNA has got a ribose sugar, DNA has got a deoxyribose sugar. They are pentose sugar. The difference is on carbon atom number 2. When you look at carbon atom number 2 of ribose sugar and carbon atom 2 of deoxyribose sugar, carbon atom 2 on the ribose sugar has got a hydroxyl, whereas carbon atom 2 on the deoxyribose sugar has got a hydrogen. So that is the difference of the two sugars. This one has, deoxyribose has hydrogen and ribose sugar has a hydroxyl. Now, when we come to the nitrogenous bases, you look at the nitrogenous bases of RNA, it has adenine, it has cytosine, and it has guanine, and then it has uracil. Those are the nitrogen bases. But when you look at DNA, DNA has adenine, it has cytosine, it has guanine, and it has thiamine. So you will see that they have a difference. The difference they have is that in RNA has uracil, whereas DNA has a thiamine. That is the only difference in terms of bases, nitrogenous bases. But when it comes to the pentose sugar, RNA has got a hydroxyl, while DNA has got a hydrogen. Those are the major differences between RNA nucleotide and DNA nucleotide. Polynucleotide formation. RNA nucleotides are linked to form a single polynucleotide strand via condensation reaction. We are saying the nucleotides, they are going to be linked together and they will form one single polynucleotide strand. And this long polynucleotide strand is going to be formed via a process or a reaction which we call condensation reaction. And a condensation reaction, it is where a water molecule is released or water is produced. Now let us look at our structure and see. What we see here, like this one here, this is a nucleotide. That one is a nucleotide. And when you come also down here, this one here, like this This one is also a nucleotide. And this one here is also another nucleotide. So the nucleotides, we are saying they are going to be linked to form a single polynucleotide. So the whole of this one here like this, all of it when it is joined together is what we are referring to as a single polynucleotide. Now, we say it is formed via condensation reaction. So what is going to happen between this nucleotide here? This is on carbon atom number 3 here. If you look at carbon atom number 3 of this nucleotide, it is going to form a bond with the phosphate here, which is on carbon atom 5. This phosphate here, which is on carbon atom 5, is going to form a bond. This one that we are seeing in red, that one is a bond which is formed between this nucleotide and this nucleotide. So that one, that bond to be formed, that red bond to be formed, we are going to lose a water molecule. And when we lose a water molecule, then we shall form that bond. And that one is what we call the condensation reaction. And then when you look at this one here, this bond here, this carbon atom number 3 here, and this phosphate, which is on carbon atom 5, they are also going to form a bond. And the bond that we are forming here is going to come from a result of condensation reaction. So which means here we shall also lose a water molecule. So that's why we are saying that the nucleotides are linked to form a single polynucleotide strand. So the whole of it is a single polynucleotide strand, which is formed via the condensation reaction. Condensation reaction results in the formation of a bond after releasing a water molecule. The 5-phosphate group of one nucleotide attaches to the sugar of another nucleotide at 3-hydroxyl. So let us try to see that one and we see what we are talking about. We are saying that the 5-phosphate, we are saying the 5-phosphate group of one nucleotide attaches to the sugar of another nucleotide, and that is at the 3-hydroxyl group. So let us look at that one and we see. We are going first of all to look at the different carbon atom so that we can know exactly where we are looking at and what we are talking about. We are saying that a 5-phosphate group. So before we go to the 5-phosphate group, let us first see on our nucleotide. This is carbon atom 1, carbon atom 2, carbon atom 3, carbon atom 4, and carbon atom 5 is here. Carbon atom 1, 2, 3, 4, and 5. Then here we shall have 1, 2, 3, 4, and 5. So we are interested in carbon atom 5. We are interested in carbon atom 5. The carbon atom 5 here. And carbon atom 3. 1, 2, 3. And this carbon atom 3. Carbon atom 5. And carbon atom 3. Now I want us to see those positions where they are. At the sugar base, that's where we have 3. I want to change the color of carbon atom 3. This is carbon atom 3. Okay, that is carbon atom 3. This is carbon atom 3 here. Are we together? And the one we are seeing in red, that is carbon atom 5. So what they are saying here, they are saying that the 5-phosphate group, meaning that it is on carbon atom 5. The phosphate if you look at our phosphate, this one here is our phosphate. The one we are seeing here, this circle, is our phosphate. And this one here is a phosphate. Even this one here is a phosphate group. Now the phosphate group is attached on carbon atom number 5. It is attached on carbon atom number 5, which is there. This one is attached on carbon atom number 5, which is there. And this one is attached on carbon atom number 5, which is there. So they are saying the 5 carbon atom phosphate group, this phosphate group is on carbon atom number 5 of one nucleotide, of this nucleotide here. This one is this nucleotide here. And we are talking about the 5 phosphate group on carbon atom number 5 of this one nucleotide attaches to the sugar of another nucleotide. Now we have another nucleotide is here. This is another nucleotide. So on this nucleotide now, we are going to attach on carbon atom number 3. Remember, this one is now our number 3 here. So when you look at number 3 here, and then here we have number 5. So when we connect those two, number 3 and 5, that is the bond that we are going to form between here and here. That is the red bond, the red line that you are seeing. It is at carbon atom number 5, where we have the phosphate group of one nucleotide attaches to the sugar. This is a pentose sugar, to the sugar of another nucleotide at carbon atom number 3. There is an hydroxyl here at carbon atom number 3 here. That's where it is going to attach itself here. So that is what we are trying to explain. The nucleotides strand will run in a 5 to 3 direction. So this is 5 here. We have a 5. And this one is a 3. Where we have a phosphate is a 5. And where we have a sugar is a 3. So it is running from 5 to 3. 5 to 3. That is how the line is running. It runs from 5 to 3. So that's why it says that the nucleotide strand runs from in a 5 to 3 direction. The bond that is formed between the two nucleotides is a covalent phosphodiester bond. That bond that is formed, the one that we are seeing in red, this bond here, that bond that is formed is what we are referring to as a covalent diester bond. It is the one which is formed between one nucleotide and another nucleotide. And that bond is the one that we call the covalent phosphodiester bond. So that bond there is what we call the phosphodiester bond. Nitrogenous bases. Nitrogenous bases. DNA and RNA nucleotides each is composed of one of the 4 nitrogenous bases. We are saying that DNA and RNA nucleotides are each composed of one of the 4 nitrogenous bases. Now these are the bases. We have 5 types of bases, but each nucleotide is having one base, at least one of them. Now there is guanine, there is adenine, there is cytosine. All the bases of DNA, the nucleotides of DNA, RNA nucleotides, they can either have guanine, adenine, cytosine. But when it comes to uracil and thiamine, thiamine is only found on DNA, whereas uracil is only found on RNA. But the other three, these other three, can be found on any of them, either DNA or RNA. These three bases can be found on the DNA, RNA nucleotides. That's guanine, adenine, and cytosine. But when it comes to thymine, thymine is only found on DNA, and uracil is only found on RNA. Now, adenine and guanine are both double-ringed, and we call them purines. That is, adenine and guanine, they are double-ringed, and we refer to them as the purine. So when you look at guanine here and adenine, they have two rings. They have two rings. That's why we are calling them double-ringed. They have this one, and then they have also that one. And that is a double ring. There is this ring here, and then there is this ring here. And those ones that have got double rings are the ones that we are referring to as the purines. And the others, they have got only the single ring. They have only single rings. Then cytosine. Cytosine and thymine, uracil, these ones are single-ringed. They are single-ringed, and we refer to them as pyrimidines. That is cytosine, thymine, and uracil. They are single-ringed, and we refer to them as pyrimidines. Here you have cytosine. Cytosine here, you can see it has only one ring. It has only one ring. Then we have thymine. Thymine also has one ring. And then we have uracil. Uracil also has only one ring. So these ones are the ones that we are calling pyrimidines. They are called pyrimidines. Pyrimidines, cytosine, uracil, and thymine, those are pyrimidines. They have only a single ringed. They are single ringed. Sorry, they are single ringed. Let's look at this DNA structure. Complementary base pairing. DNA structure, complementary base pairing. Now, DNA molecule forms a double-stranded structure. They form a double-stranded structure, which we call a ladder. That is the structure of DNA molecule. The DNA molecule forms a double-stranded structure. Now, the double-stranded structure, there is this line here. We have this side, that is one strand, and then the other one running from the other side, that is another strand. So they are double-stranded, or what we call like a ladder structure. That is the DNA. Now the sugar backbone, sugar phosphate backbones are connected by the base. Sugar phosphate backbone are connected by the base. What are we talking about? Now I want us to look here. This one is a phosphate. This one is a phosphate. This one is a phosphate. This one is a phosphate, phosphate, phosphate. And then we have the sugar. We have this one is a sugar. This one here is a sugar. This one here is a sugar. And this one here is a sugar. And lastly, this one here also is a sugar. So now when you look at them, the structure, the backbone, you will see that it is like as if the phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. It is like they are the ones that are making what we are calling the backbone of the structure. It is made up of a phosphate, and a sugar, the backbone. So the same applies this side. A phosphate, a sugar, a phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. So you find that the backbone is made up of a sugar and a phosphate on all the strand. Now, that backbone of a sugar phosphate is connected by what we are referring to as the bases. The bases here in the middle is the bases that are connecting one backbone this side and another backbone the other side. Then here we have the bases. They are forming like ladders, bases. They are connecting the sugar phosphate backbones. Then here we have the bases, and also here we have the bases. Bases are connecting to the backbone of a phosphate, sugar, sugar, phosphate, sugar, phosphate, sugar, phosphate. That backbone is connected to by the bases, and it is connecting to this sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. So the sugar phosphate is the sugar phosphate backbone, and those backbones are connected to by the bases. Now, the nitrogenous bases from each of the two strands form a complementary pairing connected by hydrogen bond. So we want to look at the nitrogenous bases, and these bases are from each of the two strands. They are going to form complementary pairing connected by hydrogen bonds. Let's look at those bases. Now, this base that we have here, that is cytosine. This base is cytosine. This base is cytosine. Now, you can see when you look of cytosine, cytosine connects with guanine. This is guanine. This one is guanine. This one is guanine. Those are the bases. Then we have adenine. This is adenine. This one is adenine. And adenine is connected with the thiamine. This is thiamine. This is thiamine. So those are what we are calling the nitrogenous bases. They are our nitrogenous bases. And those bases from each of the two strands are complementary. They form a complementary pairing. So cytosine is going to pair up with guanine. And when it is pairing up with guanine, they do using hydrogen bonds. If you look in the middle there, you are going to see the hydrogen bonds there. Those are the hydrogen bonds which are pairing. Now, when you come to adenine, adenine also pairs up with thiamine. And they are pairing using the hydrogen bond in the middle, those bases. Then when you come to guanine, here, guanine is pairing up with cytosine. Those are bases. And those bases are joined by hydrogen bond in between them. So that's what we are saying, that they form a complementary pairing. They form a complementary pairing, and that one connects them by hydrogen bonds. Cytosine pairs with guanine. That one we have already seen. Cytosine is pairing with guanine via 3 hydrogen bond. Now let us look at this one here. Cytosine is pairing with guanine. So when you look at this one, you are going to see the hydrogen bonds there. You will count it is 1, 2, 3. 3 hydrogen bonds. So that's why we are saying cytosine is going to pair with guanine, and they are going to pair via 3 hydrogen bond. And if you look at it here, you will see that cytosine is pairing up with guanine, and they are using 3 hydrogen bonds. Then we say adenine pairs up with thiamine. and they do this via 2 hydrogen bonds. Let's look for adenine and thymine. We have adenine here joining up with thymine. Now when you look in the middle, you will see that they have bonds. And if you try to count the bonds which are joining them, the hydrogen bonds there, you're going to see that they have got 2 hydrogen bonds. So a mismatch in pairing would destabilize the DNA. If there is any mismatch when they are pairing, that one is going to destabilize the DNA. So when we are matching, it is supposed to be cytosine goes with guanine, adenine goes with thymine. But you cannot connect adenine with guanine. Neither can you connect thymine with cytosine. That mismatch of pairing would destabilize the DNA molecule. DNA structure, or what we refer to as a double helix. Now, in order for 2 sets of nitrogenous base to pair, the 2 strands must run in antiparallel direction. I want us to understand this one. For the 2 sets of nitrogenous bases to pair, the 2 strands must run antiparallel direction. So here when you are looking here, we have said already that the DNA is a double-stranded. There is this strand this side, and there is this strand the other side. This strand has got bases, 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 bases. And the other strand has also got bases, 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 base, and so on and so forth. So for those bases to pair up, the strand must be antiparallel. We are saying antiparallel. So when we were looking at, we saw that we had a sugar, and a sugar, a pentose sugar. Then on a pentose sugar, we had a phosphate, like that direction. So all of them would be facing in that direction, moving upwards. Therefore, when we say that they should be antiparallel, if this strand, the sugar, is facing up, then this strand, this side, the sugar should be facing down like this. It should face down like that. That would be the sugar, and the phosphate would be facing down. This one is facing down. The other one is facing up. That is what we are calling antiparallel. So the base here will connect with the base the other side. That base there will connect with the base which is coming from this side. So here we shall have another base from this side. So we shall have another base here. So for those 2 bases to connect to each other, the strands should be opposite, in opposite direction. One, the sugar and the phosphates are facing up. Then another one, the sugar and the phosphates are facing downwards. That is what we are referring to as anti-parallel direction. So when you look at this one is facing either down, and this one should be facing upward. So it forms the 2 structure. Now here when you see the 2 structure, one strand is moving like that, going down, down, rotating like that. So you may find that that one is facing down. Then you are seeing another one. Another one is moving now up. Another one is moving up like this, moving like that, going up like that. Now that one might be facing upwards, but their bases are connecting. You can see that in the middle they have bases connecting, their bases connecting, but the strands are facing in opposite direction, which we call antipode direction. The pentose sugar, we are saying the pentose sugar The pentose sugar points in opposite direction. We have already seen that one. We are saying that the pentose sugars, they point in opposite direction. We have seen a pentose sugar, one of them is pointing upwards and another one is pointing downwards. That's what they are saying. The double-stranded molecule then it twists in order to adopt the most stable energy configuration. So when you look at the strand, this one is just showing us how they are connecting. But when it comes to the real sense, they do twist around like that. That twisting, when they are twisting around, the structure twists around. The twisting around is to make it stable. It makes the stable energy configuration. So we are saying that the double-stranded molecule then it twists in order to adopt the most stable energy configuration. So it twists around so that it can adopt the most stable energy configuration. DNA is a double helix. DNA is a double helix. This twisting around is what we are referring to as a double helix. It has two strands that are twisting around. That twisting is what we refer to as the double helix molecule, and it is made up of two anti-parallel strands of nucleotides. Two anti-parallel strands of nucleotide. So it is made up of two strands. We have one strand here, and then we have another strand this side. And this strand is made up of nucleotides. Wherever you are seeing a base, that's a nucleotide. Wherever you see a base inside, it's a nucleotide, a nucleotide, a nucleotide. So the nucleotides join together and they form what we are referring to as a double strand, a strand. So it is made up of very many nucleotides. DNA is made up of very many nucleotides. So if you look at this one, how many nucleotides does it have? This is a nucleotide 1. This one is a nucleotide 2. This is a nucleotide 3, 4, 5, 6, 7, 8. This strand is made up of 8 nucleotides. DNA forms a stable double helix arrangement. DNA forms a stable double helix arrangement. Our helix arrangement is that one that we see that goes on twisting. Is what we are referring as the double helix arrangement. Roughly 10 to 15 base pairs per rotation. Every rotation has about 10 to 15 pairs of 10 to 15 pair of bases. So they are saying in this curve, as you rotate from here like this, you come and rotate around like this. That rotation, one rotation like this of a strand, you can find there 10 to 15 base pairs per rotation. RNA structure and type. Let us look at RNA structures and types. Here we say that RNA is usually a single-stranded. RNA is a single-stranded but can form loops via complementary base pairing. RNA is a single-stranded but can form loops via complementary pairing. Now, when we say that it is a single-stranded, we saw that DNA has got two strands. Therefore, we refer to it as double-stranded. But RNA is a single-stranded. It has only one strand. And we are having examples here, like for example, messenger RNA. If you look at messenger RNA, it has only one strand. And that one strand that you are seeing, that backbone, is made up of a sugar phosphate. Then these ones that you are seeing up, which are on the backbone, those ones are the ones that we call the nucleotide, the bases. Those are bases, 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 bases. So their bases are not pairing up. They are not pairing up. So RNA, messenger RNA, is a transcript copy of a gene. That is, messenger RNA is a transcript copy of a gene, which I'll see that one later. It codes for protein. Then we have a second type of RNA. This one is a ribosomal RNA. And ribosomal RNA is a catalytic component. It is a catalytic component of a ribosome. It is involved in also protein synthesis, but we shall look at them later on. Then the third type of RNA is the transfer RNA. Transfer RNA, for it transports amino acids to the ribosome when they are going to form proteins. So those are the three types of RNA structure. We have the messenger RNA, we have the ribosome RNA, and the transfer RNA. We say the RNA, they are single-stranded, but can form loops. We are saying they can form loops via complementary base pairing. We have seen the messenger RNA, it is only one strand. And when you come to this one of ribosome, inside you can see the RNA. That RNA, now where it is bending, where it is forming loops, its bases can end up pairing up where it is forming the loops. Now when you come here, you will see now the transfer RNA. It is a single strand, but it forms loops. Now where you are seeing where it is forming those loops, it can form what we call base pairing up. Bases can end up pairing up at those points which are very close to each other. So that's why we are saying that it can form loops via complementary base pairing. Three main types of messenger RNA. We have the messenger RNA, we have the transfer RNA, and the ribosome RNA. We have seen those ones. DNA elucidation. Watts and Crick. James Watts and Francis Crick first elucidated the structure organization of DNA molecule. It was James Watson and Francis Crick that made the structure, tried to present the structure organization of a DNA molecule. They were the first one to propose the first structure organization of a DNA molecule, and they proposed that it was a double helix, and that one was in 1953. That's when they carried out that experiment. Their efforts were guided by contribution by other scientists. Though for them they elucidated the structure organization of DNA molecule, but the efforts were already there. The efforts guided them. The contribution from other scientists, it's what they used to come up with the structure of the double helix of DNA. So Pauling was one of the people who had done prior work. For him, he worked on the molecular distance and bond angle. He looked at molecular distance and bond angle, because remember we said that a DNA molecule is made up of nucleotides, and those nucleotides, they join each other using bonds. So for him, he worked on the molecular distance and their bond angles. Then there was Levin. Levin, for him, he worked on the nucleotide composition. The nucleotide is made up of what? But we know that a nucleotide is made up of a sugar, is made up of a phosphate, and is made up of a base. So it was Levin who worked out that one, that DNA is made up of nucleotides, and nucleotides are composed of the sugar, composed of the phosphates and the bases. Then Chagaf, for him, he looked at the base composition, and is the one who found out that there are equal purine and pyrimidine number. The number of purine in a DNA molecule and the number of pyrimidine, they equal to each other, they are equal. That is the composition of the bases. Then Franklin, for him, he said that the DNA is a helical structure. It has a helical structure. So all those ones, that information from the different scientists, it is what Crick and Watson used to elucidate the structure organization of DNA. Watson and Crick built a model using trial and error to quickly assess the viability of potential structure. So they constructed their model. It was trial and error. So here they were trying to carry out some work on the model, how the model should look like. They were trying it out here, but they made it helix. Helix, it was proposed by a certain scientist. They were pairing up the bases. Pairing of the bases was also proposed by another scientist. So they ended up using the scientists' ideas. They assembled the DNA model that demonstrated sources. One, antiparallel. They used the antiparallel DNA strand facing in different direction. Two, the outer sugar phosphate backbone. They used that one also. Three, the inner base pairing. Those are complementary bases. Four, the overall shape and a double helix. That's what they were using. Antiparallel, DNA strands facing in opposite direction. Outer sugar phosphate backbone, the outer layer or the outside part was containing of a sugar and a phosphate backbone. And then the inner layer was a base pairing, complementary. So what was joining the two strands was the base pairing from one side to another, and then the overall shape, which is a helical, a rotating shape. DNA organization. In prokaryotes, DNA is naked. In prokaryotes, DNA is naked, meaning that not bound to proteins. So you look at the prokaryote here, you will see that their DNA is totally naked outside. There is no enclosement. They don't have any protein enclosing their DNA. Now, when you come to also the DNA adopts a circular configuration. When you look at the structure, it is circular. If you look at it, the structure is circular. So the DNA of prokaryotes adopt a circular configuration. And then in eukaryotes, these are organisms that have a nucleus. The DNA is linear. So when you look at this one, it comes from one point and then it will end at another point. There is this point here where it is coming from, and then it will go rotate around until when it reaches the other end. So we say that it is linear, not circular. It is not connected. It's linear and is bound to histone. It is bound to histone protein. We are saying that DNA is bound to histone protein. Now, histone protein is the one which helps it in packing histone proteins to form a compact structure which we refer to as chromatins. So, in eukaryote, DNA is linear and is bound, rotated on proteins, certain proteins, okay, which we call histone protein, so that it can form a compact structure which we call the chromatin. This long strand of chromatin may further condense and become what we call a chromosome during the process of cell division. So the long strand of chromatid may further condense into a chromosome during the process of cell division. When you look at cell division, when the cell is not dividing, the chromosomes are very long, or chromatids are very long and thin. But when they are going into cell division, they condense and become short. And how do they condense? By rotating, winding themselves on the histone protein. And then they become shorter. And that becoming shorter is what we are calling condensing. They become shorter and thick. And that is what we are calling condensing. They condense into what we are calling the chromosomes. Chromosomes are the ones that we can see and use during cell division. Each chromosome contains a single DNA molecule. Each chromosome contains a single DNA molecule. So when you see one chromosome, it is made up of one single DNA molecule. Hello, my friends. Thank you very much for participating in nucleic acid. We have talked about the nucleotides, talked about the differences of DNA versus RNA, the nitrogenous bases. We have looked at the structure of DNA, types of RNA. We have seen the work of Watson and Crick, and then DNA organization. Please don't forget to subscribe.